covering organic molecules. We're actually gonna start with just chemistry. It will cover inorganic compounds too, slightly, and then the second video will be focusing on organic molecules. Okay, so let's go ahead and begin. So matter, what is matter? It's anything that has mass and occupies space. States of matter include solid, that has defined shape and volume. That's one. So here you can see example of solid matter. Liquid, when we have defined volume and changeable shape. The second picture, and gas, changeable shape and volume. So when you have a solid holds shape and it has fixed volume. Liquid, shape is, it takes shape of a container. And when we have a liquid, we have what we call free surface, right? So it will be important because when we cover tissues, we need to know what free surface is. And volume has, liquid has fixed volume. Gas take shape of a container and volume of the container. Energy. Energy is a capacity to do work or put matter into motion. There is two types of energy. Kinetic is energy of action, and potential, stored energy. So, so here you can see we divide energy into potential and kinetic, and some example of potential energy include gravitational, elastic, nuclear, chemical. An example of kinetic energy include mechanical, thermal, radiant, electrical, and sound. Chemical energy stored in bonds of chemical substances. Electrical energy results from movement of charged particles. Mechanical energy directly involved in moving matter. Radiant or electromagnetic energy exhibits wave-like properties. For example, visible light, ultraviolet, or X-ray. Energy cannot be created or destroyed. Energy can be only converted from one form to another, and conversion is inefficient because some energy in the parentheses is lost as heat. So of course we cannot lose energy because energy cannot be destroyed. We just have less energy that can be used to do some work. So here's a good example over here, how we have potential energy. It's a chemical potential energy from gasoline. And in the presence of oxygen through reactions that called combustion, this energy of chemical bonds in the gasoline is transformed into kinetic energy that moves your car, movement of your car, right? And we have waste products, carbon dioxide, and water. Why this example is important? Because if you look at the example below, we have glucose from food that has potential chemical energy stored within bonds between atoms. In the presence of oxygen, your cells break down this glucose through the process called cellular respiration. And you transform energy to make ATP. And ATP is energy for cellular work. And waste product will be carbon dioxide and water as well. And in both example, you can see that some energy is released in the form of heat that cannot be used to make ATP or cannot be used to move your car. So that's called laws of thermodynamics. Energy cannot be created and or destroyed. Energy only can be converted from one form to another. We can create ATP, right? But we cannot create energy. So composition of matter. Matter is made of elements. Elements cannot be broken down by ordinary chemical means, and each element has unique property. Physical properties are detectable with our senses and are measurable, and chemical properties, how atoms interact or form or break bonds with one another. Elements are made of atoms. Atoms are unique building blocks for each element. So each atom has atomic symbol, one that is made of 1 or 2 letters, chemical shorthand for each element. So you find all the elements in the periodic table of elements. And if you look at the example, element has a symbol. In this situation is C for carbon. It has atomic number 6 and atomic mass that for carbon is 12.01. So major element of the human body that make up about 96% of body mass are oxygen, carbon, hydrogen, and nitrogen. Lesser and trace elements of human body takes less than 5%. So if 96% are your major element, then we have less than 5% lesser and trace element. So less about 3.9 of body mass and trace element less than 0.01% of body mass, but still required for proper body function. Example, they can be part of enzymes. They can be coenzymes. So you can look at some elements over here that are lesser or trace, like phosphorus, potassium, sulfur, sodium, chlorine, and the trace element like copper, silicon, cobalt, iodine, that the total amount of trace element less than 1% in our body. 3, organization of matter. Matter can be either a mixture of pure substances. So the question you would ask to separate matter in these 2 large groups is, can it be physically separated? So if you have some amount of matter that made of, elements and atoms, if you can physically separate it, then it's going to be mixture. If you cannot, then it's going to be pure substances. Now, for mixture, the next question to ask, is the composition uniform? If it's yes, that it's homogeneous mixture or solution. If composition is not uniform, is heterogeneous mixture. Heterogeneous mixture include colloids and suspensions. And pure substances, the question to ask, can it be chemically decomposed? If yes, then it's compound. If not, it's element. So mixtures, we have no chemical bonds between components, can be separated physically, such as a straining or filtering, and can be heterogeneous or homogeneous. Compounds can be separated only by breaking bonds, and all are homogeneous. So here we have examples. We have element, that atoms of the element of argon exist on their own. Or we can have elements that oxygen atoms join in pairs. So both argon and oxygen are elements. Now, if we have mixture, we don't have chemical bonds between components. Air is a mixture of elements and compounds. And compound, we have elements that hold together, or I'm sorry, atoms that hold together by chemical bonds, but we can still break those bonds. So for example, carbon and oxygen atoms are joined together in carbon dioxide. So that make a compound. Most matter exists as mixtures, two or more components that physically intermixed. Three types of mixture include solutions, colloids, and suspension, right? So if you go back just to make sure we kind of get it. So mixture, as shown over here on this picture, when we have elements and compounds that do not have chemical bonds between them, and they can be physically separated, right? For example, if you filter it, you can separate mixture, right? So those mixture can be either solution, colloids, or suspensions. Now, if you look at the differences between, if it's a solution that we have small particle size, they appear clear, separation does not separate, passes through the filter paper, and light can pass through, for example, salt solution. If it's a colloid, then particle size are bigger, they look cloudy, does not separate, passes through the filter paper, scatters light, and for example, your mail. Suspension, we have even bigger particles, they look cloudy, separates or settles, particles do not pass through the filter paper, light cannot pass through, an example would be a muddy water. So here's some other example of solution. Solid particles are very tiny, do not settle out or scatter light. So example, your mineral water. Then we have the colloid. Solid particles are larger than in a solution and scatter light, do not settle out. Example, gelatin. And suspension. Here they give you example of blood as a suspension. So solid particles are very large, settle out, and may scatter light. Now, if you look at the blood example here, when they're talking the blood is suspension, they're talking about plasma and formed elements, or what we used to call cells. So you have plasma and you have white blood cells, red blood cells, platelets. So when you compare plasma and formed elements or cells, then it will be suspension. But blood is absolutely interesting example because blood is example of colloid as well. Because if you look at plasma itself, in the plasma, you will have larger proteins and proteins of the plasma and fluid of the plasma are in a colloid state. Now, if you look at plasma and dissolved particles in the plasma, like a sodium, like a potassium, then this will be example of the solution. So blood is a great example of all three. Solution, if you compare fluid portion, that is water of plasma, and dissolved particles, such as sodium, potassium, chloride, that's a solution. If you look at the water of plasma and larger proteins, that will be colloid. If you look at plasma and cells, then it's suspension. Solution. Homogeneous mixture, usually transparent, example, again, atmospheric air or seawater. And in solution, we always will have solvent that present in a greatest amount. Usually it's a liquid. In the biology, it's always water. And solute present in smaller amounts. Now, concentration, concentration of solution can be expressed in percent or parts per 100 parts, milligrams or deciliters per deciliter, milligrams per deciliter, molarity, moles per liter. 1 mole is atomic weight of an element or molecular weight, sum of atomic weights of a compound in grams. And 1 mole of any substance contains 6.02 multiplied by 10 in 23rd power molecules. That is called Avogadro number. So we will work with a lot of solutions. And in biology, we have many fluids of your body, intracellular, extracellular, they exist as solutions. So it would be nice to understand how we can express how much solute we have in this particular solvent, right? So we have always solute and solvents. Now, chemical bonds. Electrons are interactive. Now, outermost energy level of electron level fully occupied or contains 8 electrons. Oh, I'm sorry, not interactive, stable. So let me start over here. So when we have chemical bonds between atoms, we have actually electrons that interact, right? And when electrons interact, we call these chemical bonds. So when we have, for example, carbon and 2 oxygen atoms, and when their electrons interact with each other, carbon and oxygen form chemical bonds, right? But we have atoms that are stable and they do not interact with any other atoms. Now, why we would have this stable element? Because if you look at this diagram, in the middle, you have atom. I'm sorry, nucleus, right? So you have a nucleus in the middle, nucleus of atom, and then you have electrons, and electrons are located in the shells. Each shell has different energy level. The outermost shell, if it's occupied by 8 electrons, then this atom is chemically stable and it's not going to form chemical bond. So, for example, helium, neon, argon, right? So all this and all of these, radon even, xenon, krypton, if you look at the outermost shell of electrons, you can count. We have 1, 2, 3, 4, 5, 6, 7, 8. So each one has 8 electrons on the outermost shell, except of helium that has 2. So all of these atoms are stable. So they do not form chemical bonds, right? But if we have atoms that outermost energy level is not fully occupied of electrons, then they tend to gain, lose, or share electrons. And that's what is called bond formation. So they gain, lose, or share electrons with other atoms to achieve stability. So, for example, hydrogen, it has only 1 electron on the outermost level, energy level, and because it's only 1 level over here, To achieve stability, it needs 2. Carbon, it has 4 electrons. To achieve stability, it needs 8, right? Oxygen has 6, so it's need 2 more to be stable. Sodium, it only has 1. Now, what do you think? It's either need to gain 7, right? Or lose this 1. And then the previous energy level has 8 electrons, right? So let me kind of like go back over here. Oops. Okay, that's what I missed. I thought like, okay, it seems like I'm missing one of the slide. Now, and yes, and this is what I missed over here. So what is chemical bond? Electron occupy up to 7 electron shells that called energy levels around nucleus. Right, so here's our sodium and fluorine. Now sodium, so here's his nucleus and electrons on the different energy levels. The first level has 2 electrons, right? And the all other levels must have 8 electrons to be stable. We call it octet rule. To accept the first shell, which is full with 2 electrons, atoms interact in a manner to have 8 electrons in their outermost energy level that is called a valence shell, right? So now everything makes sense. Because next slide tell us, well, some atoms, they already have 8 on the outermost shell. So they don't need anything. They don't need to borrow. They don't need to donate. They don't need to share. They already in a perfect equilibrium, perfect energy. They have perfect stability. So they do not form chemical bonds, right? But some don't have these 8 electrons. They either have less, like well, they obviously cannot have more than 8. So they have less, but they might have 1 less, 2 less, 3 less, or they might have like 7 less. If they have like sodium, for example, only 1 electron on the outermost level, it's easier for sodium to donate this electron that then gain 7, right? When we have this process of gaining, losing, or sharing electrons, that's where chemical bonds are formed. Now, chemical bonds can be divided into ionic, covalent, hydrogen, and then we do have some other chemical bonds that you will learn in chemistry. Those are 3 important in biology. Now, ionic bond, we have metal and non-metal. Covalent, we have non-metal and non-metal, or metalloid and non-metal. So ionic bonds. Ions are formed by transfer of valence shell electrons between atoms. So here's our sodium atom that has 1 electron, valent electron. And this is our chlorine atom that has 7 valence electrons. So what's going to happen? Sodium will transfer 1 electron to chlorine. Now, when sodium loses 1 electron, it became positively charged, right? Because electrons have negative charge. So we call it cation. It's a positive charge. That means that atoms have lost 1 or more electrons. And chlorine gain 1 extra electron, so it has now negative charge, so it became anion, negative charge that gain 1 or more electrons. So we have sodium ion and chloride ions. Attraction of opposite charges result in an ionic bond. Ionic compounds, let me see, make sure I don't miss any slides. Ionic compounds form crystals instead of individual molecules, for example, sodium chloride. Covalent bonds formed by sharing of 2 or more valence shell electrons. So here we're sharing instead of donating and losing. allows each atom to fill its valence shell at least part of the time. So formation of 4 single covalent bonds, carbon shares 4 electron pairs with 4 hydrogen atoms in this example. So here's a carbon that has 4 valence electrons, and it shares those electrons with 4 hydrogen atoms, forming methane, CH4. Right, so now look, hydrogen has 2 at least part of a time. Each hydrogen has 2 electrons, and carbon has now 8 at least part of the time at its valence shell. Now, instead of drawing those electrons, in chemistry, we use just a line. So this line between atoms, between carbon and hydrogen, represent a single chemical bond. So that's a structural formula of methane. We also can form a double covalent bond. In this example, 2 oxygen atoms share 2 electron pairs. So we have 1 oxygen atom that has 6 electrons and another oxygen atom that has 6 valence electron. So they form a molecule of oxygen, O2, when they share 4 electrons between each other. Now you can see here the structural formula shows double bond between oxygen atoms. Or we can form a triple covalent bond. In this example, 2 nitrogen atoms share 3 electron pairs. So here's nitrogen atom has 1, 2, 3, 4, 5 valence electrons, and another has 5. And now they share 3 pairs. So the structural formula will have 2 nitrogen atoms and 3 lines between them. That means we have 3 pairs of electrons that being shared. Now, a sharing of electrons may be equal or unequal. Equal sharing produces electrically balanced or what we call non-polar molecule. So example is carbon dioxide. So we have carbon and 2 oxygen atoms bonded to form carbon dioxide molecules. Molecules are linear and symmetrical and non-polar. As a covalent bond can be formed when we have unequal sharing by atoms with different electron attracting abilities, and then we produce polar molecules. So example would be water. So V-shaped water molecule has 2 poles of charge, a slightly more negative oxygen end, shown here, and slightly more positive hydrogen ends, right there. And this molecule is not straight. It has like a little band over here, and this is polar molecule because oxygen is has a better, higher attraction for electrons. It has higher electronegativity. That means oxygen it's a larger atom and it attracts electrons more. So electrons spend more time around oxygen than they spend around hydrogen atom, right? And we form molecules that has slightly charged negative and positive ends. Right, so here we can put everything in a perspective, right? We have our covalent bonds over here with equal sharing of electrons, charge balanced among atoms. Example would be carbon dioxide. And then what we do actually, we moving toward the ionic bond. So we have polar covalent bond, right, instead of non-polar. So polar covalent bond unequal sharing of electrons. So slightly negative charge at one end of molecule and slightly positive charge at another end. So example is water. And when we have complete transfer of electrons, then we form ionic bonds. With separate ions, charged particles form, like sodium chloride will be the example. So really ionic bonds are just very, very polar covalent bonds when you have complete transfer of electrons. Here you have partial transfer of electrons to oxygen, right? But here you have complete transfer of electrons to chloride. Or chlorine to form chloride ion. Now, if you have slightly charged molecules, like polar molecules, like water, for example, then you would expect to see attraction between electropositive part, in this example, it's a hydrogen of one molecule and electronegative atom of another molecule. And this type of bond is called hydrogen bond. It's common between dipoles such as water, also act as intramolecular bond. So it's not bond between atoms, it's bond between molecules, holding a large molecule in three-dimensional shape. So here is how water exists, right? If we have one water molecule that is a polar, and it has slightly negative end, that is oxygen, it's form hydrogen bonds. It is a very weak bond with the hydrogen of another molecule, and so on. Now, hydrogen bonds are represented by dotted line. So the slightly positive ends of the water molecules become aligned with a slightly negative end of other water molecule. Now, hydrogen bonds are very important in biology. For example, DNA form a double helix because of the hydrogen bonds. And many hydrogen bonds also critical in the formation of three-dimensional proteins. Now, what are chemical reactions? Now, chemical reactions occur when chemical bonds are formed, rearranged, or broken. They represented as chemical equation. Chemical equation contains molecular formula for each reactant and product, relative amounts of reactants and products that should be balanced. For example, here's our reactants, hydrogen and hydrogen forming hydrogen gas, so that's a product. Arrow shows us the which way reaction goes, or hydrogen and carbon, and that form methane. So here's our product. And you can see the reaction need to be balanced. So if we start with 2 atoms of hydrogen, we have 2 atoms at the end. If we have hydrogen and carbon, right? If you, for example, if you don't have this 4 and you end up with 1 carbon and 4 hydrogen, so where this 3 extra hydrogen came from? So you need to balance. So you need to have this coefficient 4 in front of hydrogen. All chemical reactions are either exergonic or endergonic. Exergonic reaction release energy. Example are catabolic reaction. Endergonic reaction, products contains more potential energy than did reactants. So it's a store energy, and those are anabolic reaction. So here you can see exergonic reaction when we have our reactants, right? And that's the amount of energy that they store. And when reaction completed, here's our products. And you can see products have more energy than reactants. So energy is absorbed. Of course, if you're absorbing energy, you need to get it from somewhere, right? So you will use something to get this energy, and cells will use ATP to get this amount of energy that is needed from ATP. Exergonic reaction, here's your reactant and the product have less energy. And endergonic reaction, when we store energy, those are our anabolic. And when we release energy, those are catabolic reactions. Now, what is metabolism? If we're talking about all this exergonic, endergonic reaction, catabolism, anabolism, well, metabolism are all chemical reactions that take place in an organism. Metabolism is made of catabolism and anabolism. Catabolism, we already said, it's energy yelling metabolism, and anabolism is biosynthetic metabolism. For catabolism, our major goal is to release energy. Now, this energy that we will release, right? We're gonna utilize this energy to synthesize ATP, and we're gonna store this energy in ATP. In anabolism is used for growth. Now, for anabolic reaction, we're gonna use this ATP to make larger molecules, and we use energy of ADP, so it became ADP. So catabolism for energy, anabolism for growth. So anabolic reaction, the joining of small molecules to form larger, more complex molecules. So that's for growth process. Example will be dehydration synthesis reaction. So for example, we have a glucose and glucose, and we form bonds between these two molecules forming glycogen. So glycogen is a larger molecule and it stores more energy than glucose molecule. Or 3 fatty acid and 1 glycerol makes triglycerides. So again, triglyceride is a larger molecule that you synthesize from fatty acid and glycerol. So it's dehydration synthesis. Now, why it's called dehydration? Because you will release a water molecule. Or amino acid plus another amino acid make polypeptide. So polypeptide is polymer that is larger than amino acid. Or nucleotides joins together forming nucleic acid, such your DNA or RNA. So that's your building. You're building larger molecules from smaller building blocks. And to build something bigger, you need energy to do it, right? That's why anabolic reactions are actually our endergonic. Oh, I'm sorry. Yeah, endergonic. Right, because you're gonna store. So to build something, you need to use energy, right? And you're gonna store this energy in chemical bonds, right? So that's gonna be your endergonic reaction. So energy will be stored inside the products. Now, example of catabolic reaction gonna be splitting of large molecules into smaller molecules, digestion and energy production. And this type of reaction called hydrolysis. So you hydrolyze, you lysis means break down. Hydrolysis, you break down using water. Now, so now we have this glycogen and you hydrolyze it into glucose molecules and energy will be released. Triglycerides, you hydrolyze into 3 fatty acid and glycerol, and again, energy will be released. Polypeptide, you break down amino acid. That would happen when you digest food, for example, meat. Nucleic acid, you break into nucleotides. So that's all catabolic reactions that are going to split larger molecules and energy will be released. Another example of catabolic reaction is a very famous reaction, oxidation reaction. An example is cellular respiration. Oxidation, when a molecule loses hydrogen electrons and releases energy. In this example, cellular respiration, your cell's gonna hydrolyze glucose, right? Or even here, not it's gonna oxidize, apologize, it's gonna oxidize glucose because to hydrolyze it, dehydration, to hydrolyze it over here, to do hydrolysis, you need water, right? Here, you don't use water. You oxidize, you use oxygen. So you oxidize glucose. So now glucose is a larger molecules. Look at your products, carbon dioxide and water, smaller molecules. They have less energy. So that means you're going to release this energy from breaking glucose down, and that energy will be used to synthesize ATP. All chemical reactions are theoretically reversible. So theoretically, but in the biology, when reaction happen inside living organisms, we always need lots of factors to be present, like enzymes that catalyzes reactions. So many biological reactions are essentially irreversible due to an energy requirement and removal of products, right? So theoretically, they're reversible. But when we're talking about living cells, right, many will not be reversible. So either cells don't have energy to do it, or products are removed, so you cannot go backwards. Rate of chemical reactions are influenced by temperature. So the higher temperature, the higher rate of reaction. Particle sizes, the smallest are particles, the higher rate of reaction. Concentration of reactants, you have more reactants, you increase the rate of reaction. Catalysts increase the rate without being chemically changed. And enzymes are biological catalysts. So enzymes are chemicals that increase the rate of reactions, but enzymes themselves, they're not participate, they're not changed during the reaction. I mean, obviously they participate if they do something, but they're not consumed, they're not changed. Okay, now when we talked about, so let's just kind of summarize very quickly. So we started this chapter talking about matter and energy, because the whole universe around us is made of matter and it's made of energy. So we want to get at this definition. What is that matter? What is matter? What is energy, right? What is a form of energy? Can energy be created or destroyed? No, it cannot be. Now, what energy can do? Energy can be transformed from one form to another, and it can be moved from one place to another. That we can do, right? So we can convert energy. Now, okay, then what is matter? Well, matter is made of elements, right? And elements, you will find them in periodic table. Each element has its own chemical and physical properties. Each element is made of atoms, right? And so in your body, even if your body is extremely complex, right? 96% of your body made of 4 elements. That's amazing, right? Now, matter, it can be is a mixture of pure substances, and we talk about mixture such as solution, colloids, suspensions, right? And then those atoms, right? Those atoms, they can form chemical bonds between each other. Now, chemical bonds are formed because of interaction between electrons, right? And we look at 3 major chemical bonds important for biology: ionic, covalent, and hydrogen bonds, right? So, and when bonds are formed or rearranged or broken, that's what we call a chemical reaction. And in the chemical reaction, you have all these reactants and you have products. Now, all chemical reactions can be divided either into exergonic that release energy or endergonic that store energy. So another name for exergonic in biology are catabolic, and for endogonic are anabolic reaction. And when you sum all these reactions in your body, that's what you call metabolism, right? Now, we know that chemical reaction theoretically reversible, but many biological reactions are not reversible. And a rate of reaction depends on temperature, particle size, concentration. And in the biology, almost all chemical reactions require enzymes that are biological catalysts, right? Well, now when we have this reaction between atoms, right, we form molecules or compounds. Now, what are those compounds and how they can be classified? So we have inorganic compound that do not contain carbon, maybe with some exception like carbon dioxide. That's considered to be inorganic compound. For example, water, salts, many acids and bases, and organic compounds that contain carbon. They're usually large, and atoms are covalently bonded. Now, another kind of definition for organic compound would be they do have carbon and hydrogen, in organic compounds, they might have carbon like CO2, for example, but because they don't have hydrogen, they are inorganic. Organic compounds include carbohydrates, fats, proteins, and nucleic acids. Those are large macromolecules. Of course, we have many other organic compounds, and but those are specifically important for us because that's what we're going to cover in this chapter. Now, water. Water is inorganic compound. It's 60 to 80% of the volume of living cells. It's most important inorganic compound in living organisms because of its properties. So you can see it's amazing, like your brain is 83% water, kidneys 83, lungs 85, eyes 95, heart 75, muscles 75%. So if you look at your blood, 94% of water, right? Now, if you look at a female body, about 60% of female body is water and about 65% of male body is water. Properties of water. It has high heat capacity. That means that it absorbs and releases heat with little temperature change that prevents sudden changes in temperature. And you can notice it if you go to the beach, for example, and the heat, let's say it's 100 degrees, right? Fahrenheit. It's actually never 100 on the beach. Let's say it's 85, right? But you know that sand is way hotter than water. That because the water will absorb lots of heat before it changes temperature even a little. Another property is high heat of evaporation. That means to evaporate, water requires a large amount of heat, and this is useful as a cooling mechanism. So when you sweat and your sweat evaporates, you lose large amount of heat during this evaporation of sweat. Another property, it's a polar solvent molecule. Water dissolves and dissociates ionic substances, forms hydration layers around large charged molecules, for example, proteins in a colloid formation, and it's a body's major transport mechanism. So because it's solvent, it can break down crystals and dissolve them into ions in the solution, right? And water transport lots of substances through your body. It has reactivity. It's a necessary part of hydrolysis and dehydration synthesis. So over here, see when we have two smaller building blocks, and we link them together to form a larger molecule, the water is released. So that's a reactivity property. That's our synthesis, and it's dehydration synthesis. So you see how this water over here is formed, it's released, and then chemical bonds form between monomers. And hydrolysis, when we have a larger molecule, in the presence of water, this bond is broken, and we have two smaller monomers. Now, all of this reaction are not really gonna happen like this. You need, you might have some intermediate stages in between, and they require enzymes. Another property of water is cushioning, protects certain organ from physical trauma. For example, cerebrospinal fluid protects your brain and spinal cord. Okay, another important compounds are salts. So you have water, we call it water molecule, right? Salts are compounds. Those are ionic compounds that dissociate in water, contains cations other than hydrogen and anions other than OH minus, hydroxide ion. So ions are what we call them electrolytes, conduct electrical currents in a solution. Ions play specialized role in the body function. For example, sodium, potassium, calcium, and ions. So over here, you can see how those ions are important in forming a chemical concentration across cell membrane. So here's your cell, that's a cytoplasm inside. This is extracellular fluid. And you can see because we have like, so here's our sodium chloride right outside, here's our potassium inside. So we form a higher concentration of potassium inside, sodium outside, because those ions are separated. Because of the property of a membrane that we will discuss in next lectures, we can create chemical gradient for potassium and chemical gradient for sodium, and it will be important in formation of resting and action potential across a cell membrane. Now, acid and bases, both are electrolytes. Acid are proton donors, hydrogen ions. They release hydrogen ion in solution, and bases are proton acceptors. They take hydrogen from solution. Right, so for example, over here, we have a basis that would accept hydrogen forming water, right? So if you have acid, then you will increase, if acids dissociate, then you will increase concentration of hydrogen in the solution. If you have a base, the base will dissociate and accept hydrogen from solution, so concentration of hydrogen will decrease. So bicarbonate ion and ammonia are important bases in the body. Acid-base concentration. Acid solution contains a hydrogen. As hydrogen increases, acidity increases. Alkaline solutions contain bases. When hydrogen decreases, then alkalinity increases. So pretty much to understand it, like to make it simple, if you have acidic solutions, that means you have high concentration of hydrogen. If you have basic solution, you have low concentration of hydrogen. Now, pH is negative logarithm of hydrogen in moles per liter. And because it's negative logarithm, look what happened. If we have, for example, if we have a lot of, so over here, pure water, pH is neutral. pH of pure water is 7. All right, so that means it's 10 into -7 power moles per liter. All neutral solutions are pH 7. Now let's look what's gonna happen when we have acidic solution. When we have acidic solution, concentration of hydrogen ions increases, right? We have more hydrogen ions. But because pH is a negative logarithm, then pH goes down. So acidic pH from 0 to 6.99, right? pH scale is logarithmic. A pH 5 solution has 10 times more than hydrogen ions than pH 6 solution. So even with things like, oh, it's a small change in the pH 5 and pH 6, but pH equals 5 is 10 times more hydrogen than pH 6. An alkaline solution, remember, the concentration of hydrogen ions decreases, but pH goes up. So alkaline or basic pH from 7.01 to 14, right? And here we have some examples. So here's our concentration of hydrogen ions. So when it's a 10 in minus 14 power, that's our pH 14. And the pH 14 is our alkaline solution. So for example, sodium hydroxide, oven cleaner over here, ammonia, bleach, right, egg whites. And then we're moving to a neutral solution when we have our pH 7. So your blood is 7.4, so it's a little bit on the alkaline. Your milk is a little bit on the acidic. And then the concentration is actually concentration of the hydrogen ions get larger. So we have more hydrogen, right? We have more hydrogen and pH going down. So we go 6, 5, 4, 3, 2, and 1, like a wine, lemon, or hydrochloric acid with pH 0. pH changes interferes with cell function and may damage living tissues. Slight change in pH can be fatal. pH is regulated by kidneys, lungs, and buffers. So here on this example, we have our neutral solution with pH 7. Now, when we say we decrease the concentration of hydrogen, that's a basic solution. We increase concentration of hydrogen, that's acidic solution. Now look at your blood pH. Your normal pH 7.35, 7.45. If it goes to acidic this needs to be kind of changed, because here's our acidic goes over here and basic is right there. So it goes to acidic, it's called acidosis. So when you reach 7, that is really neutral pH for your blood, this acidosis. If you go to 6, that death. But remember, between 7 and 6, it's 10 times more. Over here, you have 10 times more hydrogen ions than with pH 7. And when it goes to alkaline, we have alkalosis. So if you go to from 7.45 to 7.8, it's alkalosis. Above 7.8, it's death. So either too many hydrogen ions or too few hydrogen ions can cause the death of cells and death of organisms. Now, as we said previously, we need to regulate pH by kidney, lungs, and buffers. So let's look how buffers specifically regulate changes in pH. Now, buffers are a mixture of compounds that resist pH changes. They convert strong acids or bases into weak ones. Now, what is a strong acid or strong base? When it's completely dissociated. So if you have a strong acid, we will have complete dissociation of ions. So for example, hydrogen chloride, if we have all of the molecules dissociated, then it's a strong acid. Now, one of their important buffers in your blood is carbonic acid bicarbonate system, right? In your blood, you have the carbonic acid that is a weak acid, And because it's a weak acid, it will not completely dissociate. So in a plasma, you will have your carbonic acid. It's only slightly will dissociate into bicarbonate and hydrogen. So that means, so if it would be a strong acid and you put the strong acid in a solution, you would not have acid anymore. You would only have bicarbonate and protons. But because it's a weak acid, you will have all these three compounds. You will have acid, bicarbonate, and hydrogen in your blood plasma. Now look what happened. If you have lots of hydrogen ions, so if acidity increases, then the hydrogen will be attached to bicarbonate and reaction will shift toward the carbonic acid. So you remove hydrogen ions from a solution. If you have not enough hydrogen ions, right, you have few of them, then carbonic acid will dissociate more to donate more hydrogen ions. And that's how you will keep that balance, right? So you will go this way in response to rise in pH, right? And rise in the pH, what does it mean? If pH goes above 7, 8, 9, 10, 11, if pH goes up, that means you don't have enough hydrogen ions, right? So if you don't have enough hydrogen ions, more of the acid will dissociate and you will add hydrogen ions to the solution. Response to drop in pH. Now, why pH drops? Because you have too many hydrogen ions, right? Then you need to attach them to a bicarbonate, and in response, you will produce more of the carbonic acid. But having carbonic acid in a solution is not dangerous. What is dangerous is to have these protons in the solution. That's why if you have these free protons, you will attach them to bicarbonate, you will make your carbonic acid and you're fine, right? It will not affect the pH. But the increase of hydrogen ions will increase the acidity. So that's how buffers work. And I think that's the last slide I want to cover in this lecture. Thank you for watching, and I hope it was helpful.